Hey guys, it's your boy Mike here to talk about animal meiosis today. So just a quick overview of meiosis. Meiosis is not a cycle. It is just a technique used by cells to produce gametes, which are sex cells. So since we're talking about animal meiosis today, just a couple of examples of some applications for meiosis in animals. We make gametes inside our bodies as males and females. These can be sperm or egg cells, and they can form together to form zygotes. So that's really the whole idea behind meiosis and the whole reason why it's so important. So much like mitosis, meiosis starts with interphase. So interphase is made up of three phases: G1, synthesis, and G2. For right now, we're just going to look at what the cell looks like once it's going through G1, known as gap 1. So in this phase, it's just going to grow to allow the production of more protein and give more space for the contents of the cell before division. So now, after undergoing gap 1 synthesis and gap 2 of the interphase, the cell is ready to duplicate its genetic material. And there in the center, the green stuff that's known as chromatin is about to duplicate that in preparation for meiosis 1. So basically right now the cell has enough genetic material to sustain itself according to whatever organism it belongs to. So because it has the full set of genes necessary, we call this a diploid cell. Once it replicates that genetic information, however, it will have double the required amount of genetic information and thus become tetraploid. I'm just going to take some time out now to draw some similarities and differences between the meiosis I'm describing now and the traditional mitosis cycle that we all learned probably in grade 10 science, that was. So mitosis is a cycle, unlike meiosis. So the products of mitosis can continue to divide and repeat the cycle, whereas meiosis is a specifically engineered by the cell duplication to ensure reproduction and basically just create sex cells, as I said. So much like mitosis, meiosis follows the same PMAT structure. So it starts with an interphase, but then it divides into the four main parts that we know as prophase, metaphase, anaphase, and telophase. So how about we just get into meiosis 1, the first meiotic division here, and I'll describe how there are some similarities and differences between those phases. So in meiosis 1, prophase 1 is basically like prophase from mitosis. So basically in this phase, the nuclear membrane surrounding that chromatin, or in this case we'll call it a nuclear envelope, actually disappears and leaves that DNA exposed. Once the chromatin is out within the cellular membrane there, just kind of free-floating, it condenses into small rod-like structures that we call chromatids. So in this case, they will condense into parental chromatids, paternal and maternal chromatids. So here we have some paternal and maternal chromatids scattered throughout the cell. Next, in prophase 1, they will form together to make sister chromatids. So the fathers will go with the fathers and the mothers will go with the mothers. They are joined by a small little structure called a centromere. Before we get into metaphase 1, the pairs of chromosomes will now meet. Usually a paternal and a maternal chromosome will meet up together in what we call synapsis. This will form a homologous chromosome pair. So that will be, again, one maternal, one paternal. And we can call this joining a tetrad. So once synapsis has occurred and tetrads have been formed, as you can see, there are some tips close together of different chromatids from different members of the homologous pair. Sometimes they will exchange little bits of genetic data. We call this crossing over. To commence metaphase 1, two small structures known as centrioles will appear in opposite poles of the cell. Next, the chromosomes will line up along the equatorial plane of the cell. This will prepare them for anaphase 1. Next, rod-like structures known as spindle fibers will sprout from the centrioles and latch onto full pairs of chromatids, or the chromosomes, unlike in mitosis's spindle fiber process where it tears apart chromatids from the chromosome. These will latch onto full chromosomes and commence to pull them apart. As the spindle fibers pull the chromosomes to opposite poles of the cell, this is called anaphase 1. So anaphase 1 aims to produce disjunction. So disjunction is the separation of the homologous chromosomes during this first anaphase. Sometimes non-disjunction can occur, which is the failure of homologous chromosomes to separate. That can produce a lot of uneven amounts of genetic information in the product cells, which can result in a lot of problems later when sexual reproduction occurs. Once the chromosomes have reached opposite poles of the cell, they are known to be in telophase 1. In telophase 1, new nuclear membranes will form around each of the two sets of data. Once that has happened, the cell membrane will begin to start pinching in. This is known as a cleavage furrow and will produce two diploid cells. Now that telophase 1 has completed, meiosis 2 is about to commence. So to begin meiosis 2, of course, we have prophase 2. Once again, the nuclear envelopes will dissolve, exposing the chromosomes in each of the two cells as of right now. But remember, we're not done yet. As I said, the product is 4 cells. We still have chromosomes within each of these 2 cells here, so they still need to be pulled apart into chromatids before they can become haploid gametes. 2 more centrioles will appear, 1 in each cell, and the chromosomes will line up along their respective equatorial plane in preparation for metaphase 2. Once metaphase 2 is complete, the centrioles on opposite poles of each cell will once again sprout spindle fibers that will grab individual chromatids of each of the chromosomes. During anaphase 2, the individual chromatids are torn apart from each other, exposing the centromere, which then dissolves back into the cell. The cells are in telophase 2 as the chromatids reach opposite poles of their cells. In this segment of telophase 2, nuclear membranes reappear around the sets of chromatids, and once again, the cell membrane begins pinching in and a cleavage furrow forms. Once there are 4 individual haploid cells, meiosis 2 is complete. Haploid cells means that the cell has half the amount of genetic information as a full cell has. Once again, to reiterate, to start meiosis 1, we had interphase, in which the cell replicated its data and became tetraploid. Through meiosis 1, the cell was able to break down into 2 and become 2 diploid cells. From there, meiosis 2 allowed it to break down into 4 individual cells, all of which are haploid and contain half the required genetic information. As I said, these are used to produce gametes, which are sex cells, 2 of which can fuse together, 1 male, 1 female, to produce a zygote, which has the full amount of genetic information required and is used to start new life.